you much time, but I want to tell you a short story, which is like a pandemic becomes a boon. I was also working at Bahria Town, Lahore, but due to this pandemic, I was really distressed because I thought that it is not really very enough for my degree purpose. So for the great effort by my supervisor, Dr. Muhammad Shafiq, we got the purpose of our degree, and the purpose was to write a research paper, and that was a boon for me. The title of my research paper is Genome-Wide Bioinformatic Analysis of DOF Transcription Factor Gene Family of Fragaria vesca and its comparative phylogenetic analysis with Carica papaya and Lechuga sativa. Now, here something clicks in your mind that what is DOF? What DOF actually is? So in next slide, I'm going to tell you that DOF is actually a plant transcription factor, and it's a very highly conserved DNA binding domain. And it involves in many physiological changes in our plant, like its growth, development, and various procedures like biotic and abiotic stresses. In Vitis vinifera, like in grapes, DOF is involved in bud abortion and save plant from so many stresses. In this slide, I have used that DOF is actually DNA binding domain. And other question is raised that what domain actually is. So domain is actually a segment of DNA or amino acid based DNA which has its own particular function. For moving further, I have collected so many sequences as my other batchmates told you, like from PFAM, we took the reference sequence from like genomic and CTS sequence were taken from pyrosome, and the DOF sequence were taken from prosite. And the position of DOF in subcellular level, we took that from Wolf Pisard. Here is a table in which I have shown each and everything which I have collected. The next thing was the protein alignment and phylogenetic analysis. First of all, I aligned my all protein sequence of Fragaria vesca DOFs by cluster W method. And the next thing was I have generated a phylogenetic tree in MegaX. And other was to generate the protein in ProtFrame to check that the DOF genes really exist in our sequences or not. Here is my protein sequence phylogenetic tree. The next is intron, exon, gene organization and distribution on the chromosomes. So to check that our introns and exons are present, I did analysis on F. Ginesh softberry. Now another question is raised that what intron and exons actually are. So introns are non-coded sequences, and we remove these portions during RNA splicing, and the real portion that we need are exons. So here is my other analysis, and it's tell that there are more exon related genes than intron. So my next slide is about the motifs. Now another question is raised that what motif actually is. Motifs are also the conserved amino acids, and they have some more important functions in our sequences. So I did MIM analysis on MIM suit, and here is my analysis, and they tell us, the red portion tell us that in all my sequences, we have the portion of conserved motifs. The next work I have done was chromosomal location and on the assessment of Fragaria vesca DOF genes. I took my genes through the chromosome genes from pyrosome, and I have arranged all those genes on my chromosomes. And here is M2C. I did that work. And on the, you have a look on linkage group 3, we have maximum number of chromosomes. DOF location using PFAM. So we actually want to know that DOF really exists in all my 23 sequence or not. So I started my analysis on PFAM, and I generated those pics, save each and every pic in my laptop, and then I make, on my PowerPoint, I make this table. I arrange all my DOF. On other hand, I did analysis on plant GDB gene database, which told me that what actually the function of DOF is. So they told me that in Fragaria vesca, DOF is doing helping in heat stress, is in cold stress, salt stress, in hybridization effects. And with the help of TB tool, I generated this. And the purple boxes really shows us that which DOF is doing which kind of work. Other work that I have did was cis regulatory element analysis. I put on all my each and every sequence on plant care. They send me a Gmail, and then I have saved all that data and arranged them in the form of heat map. And the best thing I have found that there was a very rare element, which was lamp element. And they were told that lamp element was responsible for light. And the lamp element was showed in FV-DOF 3, FV-DOF 18 and 20. So it was really rare element for me. I have generated this map, heat map. And the next is, the main thing is my result and discussions, genome-wide identification of Fragaria vesca DOFs. So my best thing that I have analyzed is that DOF is actually helping in structural, functional, and evolutional attributes in Fragaria vesca. I have found 23 DOF genes by phytosome, and 4 DOF genes were really important for me because they show nuclear localization signaling. And these DOF of Fragaria vesca DOF 12, 20, 21, and 22. The other thing is I have found like ranges of amino acid, pi value, molecular weight by using proteogram. And I have arranged my 7 chromosomes on all the 23 genes, and the maximum genes that I have found were 6 on the linkage group 3. Then I found like cis regulatory elements, and they told us that DOF activate in the presence. DOF is activated during the hormone, and during the activation of meristem, and during stress. And then I did the phylogenetic analysis of strawberry and try to know the orthologs and paralogs. And now I can tell that using bioinformatics, we can do a lot of.
The time shortage.
We can do a lot of. And here is my. Okay, sir. I have my last slide. Now I want to show some glimpses of my Bahria Town, Lahore. There are identifications that I did, landscapes. And here are references from my research paper. And here you go. Thank you, IAGS, for giving me a lot of opportunities. Any questions?
Thank you, Fizza. Nice job, nice work.